Meiosis is a special type of cell division of germ cells in sexually reproducing organisms that produces the gametes, such as sperm or egg cells. It involves 2 rounds of division that ultimately result in 4 cells with only 1 copy of each chromosome. Additionally, prior to the division, genetic material from the paternal and maternal copies of each chromosome is crossed over, creating new combinations of code on each chromosome. Later on, during fertilization, the haploid cells produced by meiosis from a male and female will fuse to create a cell with 2 copies of each chromosome again, the zygote. Errors in meiosis resulting in aneuploidy are the leading known cause of miscarriage and the most frequent genetic cause of developmental disabilities. In meiosis, DNA replication is followed by 2 rounds of cell division to produce 4 daughter cells, each with half the number of chromosomes as the original parent cell. The 2 meiotic divisions are known as meiosis I and meiosis II. Before meiosis begins, during S phase of the cell cycle, the DNA of each chromosome is replicated so that it consists of 2 identical sister chromatids, which remain held together through sister chromatid cohesion. This S phase can be referred to as pre-meiotic S phase or meiotic S phase. Immediately following DNA replication, meiotic cells enter a prolonged G2-like stage known as meiotic prophase. During this time, homologous chromosomes pair with each other and undergo genetic recombination, a programmed process in which DNA may be cut and then repaired, which allows them to exchange some of their genetic information. A subset of recombination events results in crossovers, which create physical links known as chiasmata between the homologous chromosomes. In most organisms, these links can help direct each pair of homologous chromosomes to segregate away from each other during meiosis I, resulting in 2 haploid cells that have half the number of chromosomes as the parent cell. During meiosis II, the cohesion between sister chromatids is released and they segregate from one another, as during mitosis. In some cases, all 4 of the meiotic products form gametes such as sperm, spores or pollen. In female animals, 3 of the 4 meiotic products are typically eliminated by extrusion into polar bodies, and only 1 cell develops to produce an ovum. Because the number of chromosomes is halved during meiosis, gametes can fuse to form a diploid zygote that contains 2 copies of each chromosome, 1 from each parent. Thus, alternating cycles of meiosis and fertilization enable sexual reproduction, with successive generations maintaining the same number of chromosomes. For example, diploid human cells contain 23 pairs of chromosomes including 1 pair of sex chromosomes, half of maternal origin and half of paternal origin. Meiosis produces haploid gametes that contain 1 set of 23 chromosomes. When 2 gametes fuse, the resulting zygote is once again diploid, with the mother and father each contributing 23 chromosomes. This same pattern, but not the same number of chromosomes, occurs in all organisms that utilize meiosis. Meiosis occurs in all sexually reproducing single-celled and multicellular organisms, including animals, plants and fungi. It is an essential process for oogenesis and spermatogenesis.